All right, so I want to talk about drug discovery this morning. Drug discovery isn't guesswork, it is not cherry picking. It's actually a process, a systematic process combining biology, chemistry, pharmacology, and even computer modeling. So, assuming you have a molecule, right, that you suspect can interact with an enzyme activities or bind to a receptor to either increase or decrease activities of a cell, or can either kill or inhibit the multiplication of a particular pathogen. So what you need to do is to go through your scientific methods, which is the ideal thing to do in drug discovery. And you know how it is. It all starts with observation, with an idea, right? So you have to put your idea to test by developing your hypothesis, designing your research work, and conducting the work. So let's go into research design and you know, execution of your research work. So you will want to test the molecule outside of the body, right? To see how the molecule fares outside of the body, literally or typically we're talking about test tubes or petri dishes, against your isolated, cells. So the process where you test your drug molecule outside of the living body is what we call in vitro. In vitro literally means in glass. And in this case, you are using a test tube or you're using a petri dish, right? So after a successful in vitro analysis, you want to check whether this molecule can actually survive or work very well in a whole system. And that's where we proceed to what we call the in vivo. In vivo literally means within a living. So you want to test your organism, or sorry, your molecule in you know, in an organism, a laboratory animals, right? It could be rats, it could be mice, it could be rabbits, it could be monkey, to see how this molecule behaves inside of a living system. And when doing an in vitro analysis, you take into cognizance the absorption of this molecule, the distributions of the molecule, the metabolism. You talk about the excretion and then toxicity. Of course, you want to check whether this molecule is toxic to the system or not. And that's where you talk about your liver function test, your kidney function test. You want to check your CBC, to see the overall activities of this molecule in a living system. Beyond that, you want to check whether this molecule can survive the high acidic content of the stomach. So in vivo, we consider all what I have mentioned in vivo. Now, there are concerns around the use of animals, laboratory animals essentially, and people are, moving away from the use of animal into a smarter model called the in silico models, right? The in silico models entails mathematical modeling, molecular docking, and artificial intelligence to predict how the molecule will behave inside a living system, right? So in this case, you are not using a laboratory animal, you are not using text tube, it is essentially computational intelligence. You use computational intelligence. Of course, after a successful in silico analysis, you want to see whether this molecule can do well in a biological system. That's what will bring us to what we call the ex vivo. Ex vivo essentially means outside of the body, but you are using living microorganisms or you are using living cells, right? So let's assume you want to test this molecule, in you want to test it on slices of, animal liver or human liver or animal heart tissues. Do you understand me? So outside of the body, actually. So you do that outside of the body, but in a living system to see how the molecule reacts, in realistic term, in biological environments. So that's what we call ex vivo. Now, ex vivo essentially bridges the gap between the in vitro simplicity and then the in vivo complexity. Now, if you are done with that, right, you will want to, you will want to, you will want to, your drug essentially, once it passes through all these experimental hurdles, it becomes what we call an IND, an investigational new drug. An investigational new drug is a candidate for clinical trials, right? A candidate for clinical trials. So once that is done, you can now proceed to clinical trials. Of course, we have, four phases of clinical trials: the phase one, the phase two, the phase three, and the phase four. The phase one essentially deals with the safety and stability of the drug in healthy volunteers, essentially. Just healthy volunteer, a sizable number of healthy volunteers where you test, the drugs to see whether it is safe and stable. And then the second phase deals with the efficacy of this drug in patients that are actually sick, in patients that have the disease. So you want to check whether this drug can actually, target your specific specific, whether it will hit on your specific target, like your enzymes, your pathogens, or a receptor, right? So phase two, of course, you have a larger number of people compared to phase one. So the phase three, it is where you do your large scale testing across communities, countries, and even continents to see how people, with different, genetic makeup and all that react to the drugs. Of course, the fourth phase deals with what we call the post-marketing surveillance. It is when you get reports after the drug has been approved, you get reports of toxicity, adverse reaction, side effects from people all around the world. So that's essentially what I want to talk about today. We've talked about in vitro model of drug discovery. We talked about in vivo model of drug discovery. We've talked about in silico model of drug discovery. We talked about ex vivo model of drug discovery, and of course, clinical trials. Thank you.